So at the outset, I must thank the organizers for giving me opportunity to present on the topic that natural products as a promising therapeutic agent for the management of the liver diseases. So the one slide I'm sharing of the institute I'm working in is the Institute of um, Himalayan Bioresource Technology, and it comes under the CSIR. And this institute is mainly working on the medicinal plant, agrotechnology, biotechnology, then chemistry, their value addition, survey mapping, digitization. So this is the whole mandate of the institute with respect to the medicinal plant. Now coming to the topic, as we know that liver is the largest in the body, and it is present in the right hand portion of the abdominal cavity and mainly supplied by the hepatic artery. The oxygenated blood is supplied by the hepatic artery, and the nutrient rich blood the liver gets from the portal vein, and then this venous blood is drained through the hepatic vein. So liver is getting the 13% of the body blood supply. So liver, as we know, it performs various functions in our body, like the production of bile and cholesterol, then specific proteins, detoxification of the drug, the toxins, then synthesis of the blood clotting factors and its regulation, processing of the hemoglobin, and conversion of the glucose into glycogen and storage, synthesis of vitamin and hormone, and involved in the various metabolic processes. So overall, liver is essential for more than 500 vital function. So the liver disease, it the 10% of the world population, and the main etiological factor, etiology which is responsible for the liver disease is the infection, mainly the viral hepatitis, then the metabolic disorders, the lifestyle, then alcohol, and the toxins and drugs. They are mainly responsible for the liver injury or liver damage, leading to the conditions like chronic hepatitis, non-alcoholic fatty liver disease, alcoholic steatohepatitis, fibrosis, and if the condition remain chronic, or it may progress into cirrhosis and may progress into hepatocellular carcinoma. So the development of cirrhosis and hepatocellular carcinoma, it may prove in the patient. So these life-threatening conditions, they are drawing considerable attention of the medical and professional scientists for the development of the therapy against these diseases. Why natural drugs are recommended for the treatment? So the popularity of the natural, as the earlier speaker also mentioned, that after COVID it is increasing. So now people are more inclined toward the use of the natural product. So the main reason they are considering the low toxicity and less side effect of the natural medicines. Then the single target or the pathway drugs, somehow they are found ineffective in preventing the diseases involving the multiple pathway or the complex pathways. So this is also one of the challenge in the drug discovery in the past decade. So over that, the natural products, they can hit the multiple target to bring out the combinatorial efficacy. And the natural products, mainly they have been applied for the thousands of the years in the traditional medicine. So people have faith, certain kind of faith over them. So if we see the scenario, the natural product accounts for approximately 30% of the clinical drugs worldwide, and around more than 65% of the global population, they are consuming the natural products. And if we see the condition in the Asian country, it is around the 80% of the population which consume the natural products. So natural products here I am mainly considering the plant origin, the secondary metabolites which the plant producing, like alkaloids or polyphenols, terpenoid, or the glycosides, and many of these compounds have been confirmed in the recent year to have a significant preventive effect on the liver diseases or the liver damage. So, if we see the condition in the liver diseases, nearly half of the agent which are used for the treatment of the liver diseases, they are either natural products or the derivative of the natural products. And the different uh, activity of these products or molecule, they are on the basis of that, these products are targeted for the various diseases like anti-steatotic or insulin sensitivity activity, like compound, they are targeting the metabolic syndrome, then antioxidant, anti-inflammatory, or anti-apoptotic compound having such type of activity. They are mainly for ALD or NFLD or the viral hepatitis. So it depends upon the activity they are showing. Now, we in India, our lab is situated in the region near Himalayas, and in Himalaya we know the rich source of the medicinal plant, very rich in the biodiversity, and over the 1800 species of medicinal value are present in the Himalayas. So we are also focusing some plant which are showing the activity against the liver diseases. So here I will share some of the example, like this is the purposes one of the plant which is present in the Himalaya from the Kashmir, my region, and this we have evaluated its efficacy against the liver fibrosis. So this Sorachia purpose, it is used substitute to the very famous plant and used in the Ayurveda also, or the traditional medicine, that is Sorachia chirata. And it is well, this Sorachia chirata is well known for its efficacy against various diseases like fever, constipation, anemia, diabetes, or hepatic disorders. But the problem with the chirata, the Sorachia chirata is critically endangered species, and the availability is an issue. So people are looking for any kind of substitute. So we have found in folklore, people are using this Sorachia parvasis for. So we made the extract out of this plant and performed the characterization of the extracts. Then we performed the in silico binding studies with the various proteins involved in the liver fibrosis. And we found that the metabolites present in the plant, they are showing very good binding efficacy or affinity with these proteins. So on the basis of these studies, then we perform the in vivo studies in the rat model of liver fibrosis, where we have induced the liver fibrosis in the rat, and we treated the animals with the various doses, and we found that the plant significantly improved the hepatic entry markers and reduced the oxidative stress in the hepatic tissue, and the plant reduced the degenerative changes induced in the liver, like necrosis, the ballooning of the hepatocytes, and the inflammation. The extent of this damage was significantly reduced by the extract treatment. Further, the fibrous tissue deposition in the liver parenchyma was found significantly reduced by the extract treatment at various doses. To confirm the effect of this extract on the various markers of this fibrosis, we studied the expression and protein level of the markers like TGI beta, alpha SMA, and we found that we effectively reduced the expression as well as the level of this marker in the tissue, and also show the anti-apoptotic activity. So overall, we found that this extract was very effective in effectively inhibit the inflammation and fibrosis development in the liver, and further it has shown the anti-apoptotic activity and prevented the liver damage. was a study published in the pharmacology. The other example in which we have studied the antifibrotic activity of the closine. Now, closine is a molecule. It is a carotenoid present in the crocus sativa, and it accounts for 10% of the dry mass. So, closine is very effective in reducing this toxicant reduce liver damage by lowering the liver injury markers, improving pathology, and reducing this deposition of fibrous tissue in the liver. And this was confirmed by studying the effect on the fibrotic marker and inflammatory factors. And overall, we found that closine restored the liver injury markers and prevent the liver fibrosis by the inflammatory and fibrogenic factor. So, were the two examples in which we found the antifibrotic effect of the molecules and extract from the of the condition of non-liver disease, as we know that this is the excess deposition in the site, which may lead to the inflammation. So NFLD is affecting around 25% of the population worldwide. However, the incidence is somewhat higher in the region around 27 to 28, or maybe in India it is up to 32%. So we evaluate the efficacy of one of the plant, which is Picoryza curva. It is a plant of Indian Himalayan region. Traditionally, the rhizome of is used to treat the liver disorder. So we evaluated the extract of this plant in the zebrafish model of the NFLD, in which we induced the fat deposition in the zebrafish larvae and treated the larvae with the extract, and found that the Picoryza curva effectively reduced the deposition of the fat in the liver of the larvae. Further, we treated the fish with the we overfed the fish to induce the NFLD, and then treated it with the extract, and we found that the extract treatment reduced the BMI in the zebrafish and reduced the expression of the lipogenic markers, lipogenic genes, the inflammatory genes, and the antioxidant gene in the liver tissue. And further, it has also reduced the fatty changes in the liver parenchyma of the zebrafish. So this just give us a clue that this extract can be used can be developed as a phytopharmaceutical for the treatment of the NFLD, and we are doing some experiment with the rodents now. So another example is the fluoretine, which is a molecule plant-derived phenolics, and it is mainly present in the apple and the apricot. So we evaluated its efficacy in the non-alcoholic fatty liver disease, and firstly we studied its efficacy in the in vitro model, where we treated the cell with the fatty acids, and then evaluated the effect of the fluoretine at different concentration. We found that the fluoretine treatment reduced the deposition of fat in the liver tissue, and also reduced the ROS generation in the cells treated with the fatty acids. And it also improved the mitochondrial membrane potential, which is one of the factor and one of the reason for the NASH development. Then further, in the mouse model, we evaluate the efficacy of the fluoretine, where we treated the mouse with the Western diet, and again treated it with the fluoretine at different concentrations. So we found that fluoretine treatment reduced the weight gain in the mouse and deposition of the adipose tissue in the body, and the fatty changes in the liver parenchyma were also significantly reduced by the fluoretine treatment. The liver injury markers and the lipid profile were also better in case of these fluoretine treated animals. The fibrosis deposition and markers of fibrosis, their expressions were also effectively reduced by this fluoridine treatment. Then we found that in the transmission electron microscopy, we found that the fluoridine induced the autophagy in the hepatocytes, and this is one of the reasons for the mechanism of anti-steatotic effect of the fluoridine in case of fatty acid treated cells. So these are the images showing the fatty changes, and these are the indicating the autophagolysosomes in the hepatocytes. So overall, we concluded that fluoridine is effective in attenuating the progression of NFLD, and the effect is produced through the induction of the hepatic autophagy. So this is the molecule which we have explored for the NFLD condition. Now, these natural products in combination, they are supposed to produce the better efficacy. So in one such study, we evaluated this synergistic effect of the curcumin and piperine against the hepatocellular cell carcinoma. As we know that curcumin is very well known compound for its medicinal efficacy. It is a curcuminoid from Curcuma longa. Then piperine is an alkaloid from the piper, and but the curcumin has having a problem with its bioavailability, whereas piperine is known as a bioenhancer. So we use a combination of these two compound, and we found that their combination produce the better efficacy in comparison to the curcumin alone by reducing the tumor development, then improving the liver injury markers, reducing the bile duct hyperplasia, the mitotic body, and then induce the apoptosis in the tumor in a more better way, and reduce the proliferation of the cells. So overall, the combination of curcumin with piperine shows the better elevation of the SCC in case of the rat model. So yes, the natural products, they have very potential in the treatment of the liver diseases, but along with their potential, there are many concerns with the natural products. And as I told earlier, that they are increasing, their popularity is increasing day by day, either due to their easy access or the cost effectiveness, or people are considering they are safe, their tolerability with the wide margin of safety. But we should not consider the natural and safety as a synonym, because the most of the product in the market, they lack in the toxicity data or the pharmacological data, and the safety of the natural products, they are mainly compromised by the contamination, the different contaminants present in the natural product, including mycotoxin or the toxic metabolites in the plants, heavy metals, radioactive contamination, pesticides. So these are and the adulteration also. So they are responsible for the mainly toxicity in case of the plant-derived or the herbal medicine. And the interactions, there is a lack of interaction studies when we are giving the combination of the herbal drug with the other synthetic drugs. So how they are interacting in the body, whether they are producing any side effects. So such type of studies are lacking. So the way forward is that strong regulations are required, and the data about the toxicity, then the adulteration, the overdose. So these are the main things which we have to take care. So in India, like when we are using as a herbal medicine, so mainly 3 category, and there are regulation, we have to follow different regulation like food and supplement or nutraceutical. So FSCCI have a very well guideline or notification in which they came up with the different plants, their doses for the development of the nutraceutical. And then when we are using the natural product in the traditional medicine, so we should follow the Ayush route. Then there is a new guideline or new class of drug, that is phytopharmaceutical, as mentioned by the earlier speaker also, that we have to identify the core marker compound and we have to follow the DGCI route for their development. So most of the natural product, they are still lacking in the clinical studies, and very less are having the mechanistic type of data for their efficacy against the liver diseases. So this is also required. Then the combination of the drugs and their mechanism need to be explored to find out a better or more effective solution for the liver disease from the natural products. So this is all from my side. I thank you all for your patience and listening.